Okay, so here we go, our next section. You guys should have finished up the first part of the organic chemistry notes, talked a little bit about your functional groups, talked about why carbon's so important and why you can have 4 bonds that go around or why you can bond on 4 things. So usually anything that we deal with organic is going to contain the element carbon, and there's always exceptions to the rule. We say carbon dioxide is one such molecule that is not considered organic even though it contains carbon in it. So as we start working through these ones that are organic, the first of the 4 that we're going to look at is carbohydrates. So remember, we have 4 types of organic macromolecules. So they are your carbs, lipids, proteins, and nucleotides or nucleic acids. And that's kind of the breakdown of how you guys are going to see them within this note section. So as we go through these, you're going to see it in this form. We're going to kind of let it sink in just a little bit. You'll see some handwritten notes. You'll definitely see some diagrams that we'll be going through as well. You guys will be doing some flashcards along the way. We have another gel lab that we want to do. There'll be some worksheets to help us out. And as we go through these notes, keep a list of questions. Hey, I don't really understand this. Can we go back through this particular part? Because this is really that building block of our particular section that we're going to be looking at. Okay, so your carbohydrates. First year bio, we talk about them. You guys did a little food lab. You broke things down. Benedict solution, burette, things like that. Within your urine lab, we use Benedict solution to check for glucose. And glucose actually is a form of carbohydrates. So what carbohydrates do within the cells, it is your quick energy source. It's the first place that your body wants to go to in order to break down food to get ATP. It wants to break down those carbohydrates. It's the best energy source for your body. If you guys remember from first year bio, these carbohydrates usually end in OSE or OS, glucose, galactose, fructose, sucrose, agarose, which is what your gel electrophoresis gels were composed of. I said it was a large sugar gel that kind of we melt down with some buffer and we pour it in and we make molds from it. Within your carbohydrates, we can then break them down into smaller groups as well. So if you're looking at words like monosaccharides and disaccharides and polysaccharides, and they're kind of confusing you, break the words down. Mono meaning 1, di meaning 2, poly meaning many. Saccharide or sacchara meaning sugar. So we're dealing with one sugar, a single sugar, or simple sugar. Your formula for a monosaccharide is C6H12O6. That means we have 6 carbons, 12 hydrogens, and 6 oxygens. They are usually going to be in a 1 to 2 to 1 ratio. For every carbon, you'll have 2 hydrogens and 1 oxygen there. Some other ways that you can tell a carbohydrate or a sugar, especially a simple sugar or single sugar, is besides that ratio that you see there, they're typically in a ring structure. So you will see these little rings right down here. Let me get you guys a little pointer going on. So you'll see right here, that's a little ring structure. There's no little ring structure. There's no little ring structure. So what we're looking at with these particular ring structures, another new word for you guys is isomers. Those structures you see there, glucose, fructose, and galactose. Glucose is kind of like a cane or a beet sugar. Fructose is this fruit sugar, and galactose is a milk sugar. They all end in OSE. They are all in that ring-like structure there, but they also have the same formula. Whether you're glucose, fructose, or galactose, you all have the formula C6H12O6. That's why they're called isomers. So they have the same chemical formula, this C6H12O6, but if you look at them structurally, the way that they are arranged, they're all a little different. And because of that arrangement that they have, it makes them slightly different simple sugars. So isomer, once again, same chemical formula, different structural formula. And what we can now do with these particular monosaccharides is that we can start to join them together and we can create disaccharides. So I can take 2 of my monosaccharides, join them, and make this 2 sugar molecule. So disaccharide 2, my formula here, C12H22O11. So you're going, well, if I had C6H12O6 and C6H12O6, how come I'm not at C12H24O12? Because that would be double if I put the 2 of them together. But remember, in order to join these molecules together, we've talked about this word before, we have to go through a process called dehydration synthesis. Synthesis means to create. We're going to create a molecule. We're going to make a molecule. Dehydration is the way that we're doing it. We need to take out water. And that's why my formula is not quite double. I am missing H and 2 oxygens there. I'm missing a molecule of water. And that was taken out in order to create a bonding spot for those 2 monosaccharides together. Okay, this is an anabolic reaction. It is a building up reaction. And these terms are going to be the same for your carbohydrates, for your lipids, for your proteins, for your nucleic acids. Dehydration synthesis is going to build up. We have to take out water. Hydrolysis means that we're going to break them apart and we're going to add some water in there. So your disaccharide is still going to be an energy source. It's a little bit bigger molecule that your body breaks down. It's going to give you a little bit more energy in the long run. Just like your monosaccharides, glucose, fructose, and galactose, I can take them and start adding them together. So if I take glucose and fructose, so let's take this structure and this structure. They're both C6H12O6 plus C6H12O6. I make a disaccharide called sucrose. Sucrose's formula, C12H22O11 plus some water that I had to take out of there. Sucrose is just plain old table sugar that you find on your table, you put in your coffee, maybe put a little bit on your cereal in the morning. If I take glucose, this molecule here, and galactose, this one here, they're both C6H12O6, join them together, I create lactose. Lactose is a disaccharide. It is a milk sugar. A lot of people have heard this before. If you are lactose intolerant, your body cannot break down this disaccharide at a quick enough pace where it doesn't make you sick. You don't have the enzymes to help speed up this reaction. Your body still breaks down lactose, but it takes too long and tends to upset your stomach. So maybe you take some sort of enzyme medication or some kind of lactate and you put it into your body. It's basically the enzymes that help to speed up the reaction so that you can break down this milk sugar into glucose and galactose. And while you're doing it, you're going to get energy from it because anytime you break a bond, you're releasing energy. Anytime you make a bond, anytime I put glucose and galactose together to create this larger molecule, I have energy stored in that bond now. Okay, that's my potential energy. When I break the bond, it's my kinetic energy that gets released. If I take glucose and glucose, C6H12O6 plus C6H12O6, I create a structure called maltose. Maltose is just a malt sugar. Find it like within candies and find it within soda. So where glucose, galactose, and fructose were all isomers because they were all C6H12O6, I can also say my disaccharides, sucrose, lactose, and maltose are also disaccharides. Are also isomers. They are all C12H22O11. But if you draw them all out, they're all structurally a little different from one another. So my monosaccharides are isomers, my disaccharides are isomers, even my polysaccharides will be isomers. So we just went through the two processes and talked about them a little bit. Let's look at them again. We did them with our Chapter 1 notes. You did them in first year bio. We'll keep talking about them. You will see a compare-contrast chart that you guys will be asking for some of these terms. Dehydration synthesis. Everybody kind of reverses these around. They think if you dehydrate something that you're kind of breaking it apart. Think about what the words mean. Synthesis means to put together. I'm doing this by dehydrating it. I'm removing water to bond two molecules together. It's kind of like if I take A plus B, join them together, and I create this larger molecule, AB. Plus, I took water out, and I have to show that water that I'm taking out of there. We need that formula to be balanced on both sides. Think about this way. I'm taking little Legos, Lego 1, Lego 2, Lego 3, Lego 4, and I'm starting to put them all together. This is an anabolic reaction. It is a building up reaction. I need to take out water for this to happen. As we go through, and we want to reverse that reaction, hydrolysis. So if I want to break down that milk sugar, lactose, I can go through hydrolysis. I drank some milk, I ate some ice cream, something with some kind of dairy product, cheese in it, and my body's going to start to break it down. So my body's going to add water to it. It's going to break the bond, so I split it apart. I take that larger molecule and I break it down into two smaller molecules. So it's kind of like taking that Lego house that you just built and now someone comes along and kicks it all over the place and breaks it down into little itty bitty pieces again. That's called a catabolic reaction. First year bio, maybe you took that cracker and you put it on your tongue and you let it sit there. That was a really big carbohydrate. Well, after a while, what happens? It kind of turned to mush. You're breaking that carbohydrate down into smaller and smaller and smaller and smaller sugars along the way. And it gets really soft and breaks down. That is hydrolysis. You're taking water from your body to break that bond. So we take AB, large molecule, add water to it, breaks the bond, and now we have smaller molecules A and B. So I can take maltose, add a little water to it, I end up with glucose and glucose. That is basic digestion. It's also a catabolic reaction. Now let's keep going. So we have monosaccharides, we have disaccharides, we have polysaccharides. Polysaccharides, poly meaning many. Sacchar or saccharide means sugar. Here my formula, I can't say, oh, it's a 1 to 2 to 1 ratio, because as you add more and more sugars to it, and we're taking out more waters to do that, H2O, it becomes a little funny. It's not exactly a 1 to 2 to 1 ratio. It's going to be close, but it's not going to be exact. So usually we just say that our ratio is C12H22O11 to that X variable that you see here. That variable is how many monosaccharides or disaccharides do I have joined together. Okay, that means that's my ratio depending upon how many sugar chains or sugars are in that particular chain. It's not going to be exactly a 1 to 2 to 1 ratio. First year bio, you tested your polysaccharides using iodine. And that iodine was kind of an orangey brown color. Test the cracker, you put it in there. Crackers, a lot of polysaccharides, it turned black. So when polysaccharides come in contact with iodine, they will turn dark black. And what a polysaccharide is: starch, complex carbohydrates, glycogen is how we store it within our liver. We'll talk about cellulose within plants. So they're all a little different depending upon the organism that we find them in. But many, many sugars, every time we add another sugar to it, we need to take out another water, which is why our formula is not exactly a 1 to 2 to 1 ratio. So let's say you have this huge pasta meal right before bed. Well, your body's like, oh, I got all this pasta. I can break it down and get tons and tons of energy, but you go to bed. And you don't really need a lot of energy when you're sleeping. You need a little bit of energy to keep everything moving and reactions occurring. But you have all this excess polysaccharides. So what your body does is it saves them as a form known as glycogen. And glycogen is how humans or animals store their excess glucose in their liver. So they take all this extra sugar, glucose plus glucose plus glucose, all these little itty bitty monosaccharides, and it joins them together to make this giant polysaccharide. And then we store it away in our liver for when you need it. So you get up the next morning, you're running late for school, you miss the bus, you're getting to school late, you forget to eat breakfast. And you're like, oh, I didn't eat breakfast. Now what am I going to do? Your body has that storage of energy. So your body starts to release this glycogen from your liver, little pieces at a time. And it starts to add water to it, hydrolysis, and it's breaking that chain. And it's allowing your body to utilize the glucose that's being broken off of that glycogen to make energy through that process of cell respiration. Plants don't really do this. So they don't store their excess polysaccharides as glycogen, but they do store it as cellulose. And this is a structural polysaccharide for the plant because they store it in a couple different ways. Plants have cell walls. We do not. So we don't need this rigid cell wall around our cells. We don't have it. Plants need this really rigid structure, and that's what cellulose is. So as that plant makes its sugars, it needs to store it someplace. And one of the ways is they'll store it as cellulose within those cell walls. So it is a structural polysaccharide. Cell walls can only be broken down by certain organisms. So termites think they eat wood. They need to be able to get energy from that particular cellulose or that plant-like structure. So termites actually have little bacteria within the gut, symbiotic relationship. They live inside the termites, and they're helping to break down that cellulose within the wood so that the bacteria can get food from it, but also at the same time, the termites are getting energy and food from it. Same thing, if you ever hear that cows chew their cud, so basically they're eating grass. If you guys ate a bunch of grass, you would be passing a bunch of grass right through it. Your body can't break it down. So the bacteria within the cow's gut, they swallow the grass, mixes with bacteria. They actually will spit it back up again. Cows have multiple stomachs. We've heard that before. They actually will spit it up again. And now they're chewing the bacteria and they're chewing the grass around, and they're letting that bacteria get in there and break down that cellulose so that the cows can get energy from the grass. And the bacteria within the stomach, they have a nice little happy home to live in, nice and warm, it's protected, and they got energy. They got food from the grass that the cows are eating. So some molecules are a little bit too big, and other organisms have to rely upon things like bacteria to help them break them down. So cellulose is a structural polysaccharide found within plants. Plants can also use it as storage. So think like potatoes will store some of their sugars, excess sugars, as starch. So we know potatoes are extremely starchy. They have these big starch granules down in the potato themselves. You may have looked at it first year bio, put some iodine on it. You saw these big black spots all over the place where they stored their excess sugars. They can also store it as something called amyopectin, depending upon the plant that we're looking at. So amyopectin or pectin is also a storage polysaccharide. So as the plant makes its glucose, it's going to store it as this very large polysaccharide known as amyopectin. It is actually a thickening agent for jams and jellies. If you have a parent or a grandparent that does canning of jams and jellies, and they take all the fruits and they're cooking them and mashing them all together, they need to add this amyopectin to make it kind of a thickening agent, to make it this jam and jelly consistency before they go through and finish the canning process. So you can actually purchase that in the store to add to the fruits that you want to go through and make into jams and jelly. Chitin is also a structural polysaccharide. So chitin we find in arthropods. We also find it in fungus. So arthropods don't really have cell walls, but if you think like lobsters, crab, shrimp, insects, they have exoskeletons. Our skeletons are on the inside of us. We have an internal skeleton. But bugs and crabs and lobsters and shrimp and all these other organisms that are within this arthropod phylum, they have an exoskeleton. So when they grow, they have to molt. If you guys ever had like a little crayfish in your fish tank before, it looks like a little itty bitty lobster. Well, as they grow, they actually will kind of pop out of their shell so that they're able to grow. And for a short time, they're very soft-bodied until that excess chitin hardens around them. If you've heard of soft-shelled crabs before, they're kind of a delicacy. They're only in season certain times because when those crabs are growing. So those crabs will kind of pop out of their shells because they're becoming too large. They kind of break right out of them. And then they have to go kind of hide for a little bit, a week or so. And during that time, their excess polysaccharides will harden around them, and they'll make a new shell around them so that they're protected. Fungi, it is not a plant. It is a heterotroph. They're going to feed off things within the ground. However, they do have cell walls. So those cell walls are made of chitin, not cellulose, like what we find within your plant cells. So how could I draw a diagram of it? Well, here's a monosaccharide, 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 and so on. Every time I join a ring to it, I need to take out a water. So I'm taking out H2O, taking out another water, taking out another water, taking out another water. So my formula depends upon how many of these are actually joined together. And the type of polysaccharide that I'm dealing with not only depends upon the number of bonds that I have, but the way that those bonds are arranged. We'll see some bonds that will go up, we'll see some bonds that will go down, and we talked about these isomers before. Same chemical formula, different structural formula. If I change the way that those bonds are going, I'm changing that particular molecule. Okay, so we'll talk about a lot of these within your diagrams. We'll see these again. Okay. If we keep moving on, so I'm going to look at this next type. So our next type is lipids. And the reason I'm going to do lipids and carbohydrates together is because they're so similar to one another, I guess we could say. They contain the same elements, carbon, hydrogen, oxygen. That's it. But unlike your carbohydrates that are in that 1 to 2 to 1 ratio, or all but in that 1 to 2 to 1 ratio, your lipids still in this carbon, hydrogen, oxygen ratio, but now we have very few oxygens. So you might see things like maybe 50 hydrogens and a whole bunch of carbons and maybe 3 oxygens in there. Not even close to a 1 to 2 to 1 ratio. And just when you see that very few oxygens there, you know you're going to be dealing with a lipid. And we'll talk about why there's so few oxygens here as we move on. Don't get scared by this diagram yet. Okay, we'll get there. Your functions with our carbohydrates, we said they are your primary energy source. Your lipids are more like your energy storage. Think about bears that have to go and hibernate. They build up all their lipids, they get nice and fat, they go off and hibernate, which means that they're slowing their body functions down and they're basically sleeping throughout the winter. It's the lipids that they're going to be feeding off of. It's the lipids that are going to be broken down to give them that energy that they have. At this point, their carbohydrates are all gone. Now they go to those lipids. Once we burn all of our lipids, we burned all our carbohydrates first, we burned all our lipids, then you start burning your muscles, you start burning your protein, and that is not good. That's not what you want, because a lot of your organs are supported by your muscles, and a lot of your organs are muscle-based, and you actually start breaking them down, and that is bad, bad, bad once you start getting to that particular point. So we'll do carbohydrates first, then we utilize our lipids, then we'll utilize our proteins from there. So they are energy storage. They also protect us. So if you think like your kidneys itself, your kidneys are, if you think about your backside, right low on your backside here, if you ever like messed around with someone and they come up and they give you a kidney shot and they kind of hit you along that backside, they're not really well protected back there. Okay, so you got your rib cage that's protecting your heart and your lung and all your other organs. A lot of them are very well protected. Kidneys are not. But what they've done evolution-wise, because we, evolved to realize, like, hey, this is not protected. Our body has put this big layer of fat around your kidney, so it kind of gives it kind of like this little shock absorber if you fall, or it's going to help protect that particular organ. And a lot of your other organs may have this small layer of fat that goes around them as well. It's also used for insulation. Your nerves, so nerve impulses, and we'll talk a little bit later on in the year about some different disorders, genetic disorders that deal with the nerve and the what's known as like a myelin sheath, kind of like this lipid layer that goes around it that's kind of missing and doesn't allow for nerve impulses to go back and forth. It will also waterproof, so ducks that you see swimming in the middle of the wintertime and you're like, how do they swim that really cold water without getting that water down on their skin? They have lipids on their feathers, so it kind of forms like this little raincoat that goes over them, and the water can't get down to that surface layer and change their body temperature. So a lot of different functions for your lipids there that we see. All right, let's look at this word, triglyceride. Tri means 3. Glyceride is what we're going to be looking at. So this entire molecule that you see here, this entire molecule that you guys see here, this big giant structure is going to be a lipid. Huge. And I can make it bigger and I can make it smaller, but it's all going to have the same parts to it, just so that you guys are aware. So what a triglyceride is made up of, it is made up of a glycerol molecule. Glycerol molecule is C3H8O3. This is a glycerol molecule right here. There's my 3 carbons. 1, 2, 3. C3. It's always that structure. I always have 4 things bonded around my carbons. So there are my 3 carbons, hydrogens, and then I have my OH groups there. So OH group, OH group, OH group, which is a hydroxyl group. When you see OH like this, that oxygen is bonded to the carbon and it is bonded to hydrogen. Remember, it's got needs 2 more in the outer ring. So it will bond to hydrogen and carbon. By it doing that covalent bond, it is stable. To this glycerol molecule, we want to add 3, that's where the tri comes from, fatty acid chains. Fatty acid chains are just these really long hydrocarbon chains, and there's really no set number for them. A hydrocarbon chain, carbons and hydrogens, you'll see it right here, carbon, 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 carbon. Remember, carbon's got 4 bonds around it. So this particular carbon that you're looking at right here is bonded once to here, hydrogen, hydrogen, hydrogen. I can keep making this hydrocarbon chain, carbon, 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 all the way through. What you're going to notice on this particular chain is that all my carbon to carbon bonds are single bonds. Because of that, that is a saturated fatty acid chain. So saturated fatty acid chains only have single bonds. Let's look at my bottom one here. Hydrocarbon chain, carbon, 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 all the way through. But then I have this double bond right here. Okay, what you're going to notice, because it's a double bond between carbons, I do not have hydrogens bonded to them at the top. I still have 4 bonds around them. So let's take this carbon here. 1, 2, and then it's bonded twice to the carbon next to it. 3, 4. There is no spots for hydrogens up here. So we're going to have less hydrogens in this hydrocarbon chain. So if you go back to the alkanes, alkenes, and alkynes, and why those formulas were a little different from one another. One was +2, one was nothing, and then one was -2. It's because the more bonds you have between carbons, the less hydrogens you're going to have. On this hydrocarbon chain, for fatty acids, at the very end is this carboxyl grouping. It was one of our functional groupings. It is a carbon double bonded to oxygen with an OH group. All my fatty acid chains have it. So it's a carboxyl group with a hydrocarbon chain to it, only carbons and hydrogens. This is the part of the chain that can vary. It can have different numbers. Maybe there's 50, maybe there's 100 carbons, maybe it's double bonded, maybe it's a single bond. That's the part that's going to change and make a slightly different lipid for us. Here's the big thing. we can go through dehydration synthesis. I need to join together glycerol and 3 fatty acid chains. I need to join all those pieces together in order to make this really big lipid. Well, if I have 4 pieces, glycerol and 3 fatty acid chains, I need to take out water. I need to take out one water for each chain that I'm adding on. So I'm losing a hydroxyl off of one and a hydrogen off the other. There's my water. I'm taking it out. I now have a new bond between carbon and oxygen. So can I take the hydroxyl off of here and the hydrogen off of here? Sure, it doesn't matter as long as I'm taking out water. So I'm taking out another water right there. Taking out a third water right there. Whenever you make a triglyceride, and I'm adding 3 fatty acid chains to a glycerol molecule, I need to remove 3 waters. You're going to notice this bottom chain that we talked about with my double bond here. As soon as you see at least one double bond between carbons, and I say between carbons because this does not count. This is not making it saturated or unsaturated. It has to be within that hydrocarbon chain that you guys see here. This is my functional group. He doesn't count. As soon as you see a double bond, at least one double bond or triple bond between carbons, it makes it an unsaturated fatty acid chain. Okay, so I'm taking out my 3 waters. I'm making this giant structure that you see here, which is going to be my triglyceride. Differences between saturated and unsaturated fatty acids. Your saturated fatty acids, we said, are all single bonds. They are solid at room temperature. So I say, think about that big steak that maybe you're going to have for dinner, and mom pulls it out and she lets it sit there and thaw it. Maybe it was in the fridge, letting it get a little bit to room temperature first. Maybe it was in the freezer, and it's got that thick layer of fat around it. That fat is your saturated fat. Butter, saturated fat. It is solid at room temperature. It doesn't melt. Lard, if anybody, I know it's a little bit old school cooking with lard, but people used to cook and bake with lard. These types of fats, these saturated fats that have all single bond between carbons, they are actually harmful for us if you eat too much at high doses. So you don't want to eat too many saturated fats. They're usually the ones that taste a little bit better. The reason they're harmful is that they can lead to something called arterial sclerosis. So they actually will build up within your blood vessels, those little fat clusters. And they can start causing what is known as hardening of the arteries, and they start to block off arteries so that blood flow can't get through. It can lead to heart attack. It can lead to stroke. Through time, we've started to, I know like from my grandmother's days, my parents' days, they love to cook with butter, where we're more like, hey, let's cook with olive oil or sunflower oil or avocado oil and those sort of things. And the reason is because they are unsaturated fatty acids. They have at least one double bond. They're liquid at room temperature. So when you leave them out and let them sit, they are liquid. Olive oil, corn oil, sunflower oil, they are your healthier fats for you. We can then change this particular molecule. So this is my fatty acid chain, my triglyceride. I can then change it. I can make something called a phospholipid. You're like, I never heard that. Yes, you have. Phospholipids make up your cell membrane. Okay? Every living thing has a cell membrane. Phospholipid bilayer. So we're only going to look at this particular structure here, which is my phospholipid. Get a little highlighter here. So this phospholipid that we have here, phospholipid bilayer. You've heard that term before. So if I think like cell membrane, this is actually what it looks like. And we're going to come back and really go through this. We're probably doing a real actual physical meeting in class and online with you guys where you're not listening to videos so that I can specifically answer questions. So if I take this particular molecule that you guys see here, because I don't think I give you one, nope. If I take this molecule here, this triglyceride, if I switch one part of it, I still have a glycerol. I still have 2 fatty acid chains. If I pull this first fatty acid chain off, and now I put a phosphate group on, that was another one of our functional groups. So glycerol, fatty acid chain, and a phosphate group instead. That's what's going to make my fatty or my phospholipid. So this single phospholipid right here, there's the little tails that you see. These little tails that you see on this single molecule are those fatty acid tails that you see, or those fatty acid chains up above. This little head or circular region that you guys see there is going to be that phosphate group. Your cell membrane is a bilayer. So we have two of these phospholipids coming together. These tails, these fatty acid tails, if you guys know anything about fats, is that they hate to be near water. If you ever made pasta, you have this pan of boiling water, you put a little bit of olive oil or a little bit of oil in it so the pasta doesn't stick together when you put it in there. That oil just kind of clunks together. It doesn't want to mix with that water. It is said to be hydrophobic. So when we talk about fatty acid tails, they hate to be near water. Well, the only way that your cell membrane can kind of stop them from being near water is creating this bilayer. So what you guys are going to see is that our tails hide out in the middle. They're right here. I have two layers. So I have tails facing this way. I have tails facing this way. It is a bilayer. I have water around the outside. I have water on the inside of the cell. My tails don't come in contact with it. They are said to be hydrophobic. The little head regions that you guys see here. So these little heads that we see right here, right here, right here. There's my first one. Here's my second bilayer. So here's 1, 2, 1, 2. Outside, inside, tails are meeting up. Those phosphate heads love to be near water. They are hydrophilic. Love, love, love water. So those little pink ones are my phosphate groups. Those little tails in the middle are my fatty acid tails. They're hiding out away from the water. I've created that phospholipid just by taking off one of my fatty acid chains and putting a phosphate group there instead. I can do the same thing with a glycolipid instead of a phospholipid. Let's keep our glycerol. Let's keep two fatty acid chains. And instead of this third one, instead of putting a phosphate group there, let's put a sugar there. So if I have glycerol, two fatty acid chains, and now I have a sugar grouping, that is now going to give me what is known as a glycolipid. And we'll talk about glycolipids a little bit more as we get into that next little set of notes that you guys will see. We'll kind of expand upon this, kind of keep this in your head, and we'll really break this structure down a little bit more. Hydrophobic, hydrophilic, same terms that we just looked at here, whether you are water-fearing or water-loving. And those tails are said to be non-polar. They are fatty acid chains. Those little tails hate to be near water. That's why they're stuck on the inside here, because water's on the inside of the cell, protected by the phosphate heads. It's on the outside of the cell, protected by the phosphate heads that love water. They are hydrophilic. Those phosphate heads are polar, and they are water-loving. Last little bit for your lipids, cholesterol and other steroids. Some of your hormones are also lipid-based. So when people go on diets and they cut out fats and they cut out carbs and they get really crumpy, well, they're messing with some of their hormones, because some of their hormones are actually lipid-based. Cholesterol. Cholesterol is actually synthesized, which means that it's created in your liver and it's found within the cell membrane. So not only do you have phospholipids, but you're also going to have cholesterol embedded into those cell membranes. So you might have this big chunk of cholesterol kind of embedded right in there. You might have another piece of cholesterol kind of put right in there. Okay, so it is embedded into that cell membrane. So you may have heard the term like your cell membrane is a phospholipid bilayer. It's fluid mosaic model, which means it's got a bunch of different pieces in there. You have little carbohydrate receptors, you got some cholesterol, you got phospholipids. It's made up of all these little pieces kind of put together there. So two types of cholesterol that you have there. Your HDL, high density lipoprotein. This is your good cholesterol. So when you see like those Cheerios commercials and, they're saying, oh, we want to lower your cholesterol, so eat these Cheerios. It is your LDL that we are trying to lower. Low density lipoprotein is your bad cholesterol. So as you get older and you get your blood checked, you get these two levels, and you want that HDL to be kind of high because you do need a certain amount of cholesterol within your body. It helps make your cell membranes. It helps with some other things within your body as well. Your LDL, we want to limit that, because once again, that can also lead to some hardening of the arteries as well. This is your steroid nucleus. You guys are going to see this when you guys get to your flashcards. So steroids such as testosterone and estrogen, male and female hormones, they all have this basic steroid nucleus. It is made completely of carbons and hydrogens and oxygens, because that's what lipids are composed of. Also, other little portions that you'll see off of here, and we'll talk about those structures a little bit more in the next grouping. But know that cholesterol testosterone, estrogen, which are steroids, some of your steroids and hormones, are lipid-based as well. The last little grouping that we have here is waxes. So waxes are more like your protective lipids. Plants, if you think about leaves, they actually have a protective wax coating on them. You can kind of scrape it off on a lot of your leaves. It's going to protect them from dehydration, especially during the warm months. Think about plants that are more desert-based. They might have a higher wax content on the outside so they don't dehydrate. Your earwax, it has a purpose. It's protecting your ear canal. Beeswax, blubber, lanolin that we find in wool of the sheep are all protective waxes that organisms are able to create. They are all within that lipid category. So we will see this again. I just kind of want to put it in the back of your head, and we're going to kind of go through a lot of the notes, and I'm going to try to hit you guys up with live stuff within class so that you guys can really start asking questions. You guys will see a set of handwritten notes. Okay, so as we're going through this, highlight stuff, go through stuff, write down questions so that you can ask me, please, please, please. Because I know you guys do in first year bio, but I also know that you don't remember a lot of stuff from first year bio. So we kind of want to just stop it right there. Okay, so we'll talk about proteins and nucleic acids during our next one. Get you guys started on your flashcards from here. And that should basically kind of get us going. Okay, so questions, please let me know. We can Google Meet, Google Chat, go through this. We'll look at handwritten notes. We'll do little practice tests and all that other good stuff.